Good afternoon, everyone, or good morning, depending on where you're calling in from. This is George Yorling. I'm Director of Medical and Scientific Affairs here at HDSA, and I'm excited to introduce our speakers today, Dr. Holly Kordasiewicz and Christina Lemonitis from ISIS Pharmaceuticals. They'll be speaking to us today and providing an update on the antisense oligonucleotide therapy for the treatment of Huntington's disease. Before I introduce our speakers, I just wanted to give us some rules for today's presentation. At the end, everyone will be muted, and at the end we'll be addressing, or the researchers from ISIS will be addressing your questions. You can ask your question at any time, and you can do so by typing it into the box highlighted here in red on your GoToWebinar menu. And just click the send button, and we will be able to get it here and distribute to ISIS. I'll just remind you that we'll be recording this for those that you may know that want to hear this, could not join us today and want to hear it at a later time, or you wanted to listen to it again. They can all be found on our website in the research section on www.hdsa.org research webinar. And I'll just remind you, we've got a number of upcoming webinars planned. In April, we'll be hearing from Dr. Jenny Morton from the University of Cambridge, where she'll be speaking on the topic of sleep and circadian dysfunction in Huntington's disease. And in May, Joe Giuliano, who's the Director of Clinical Operations at CHCI Foundation, will be providing the community an update on Enroll HD. And I'll just point out that we do have an email address set up, researchupdates@hdsa.org, where if there's a webinar topic that you'd like to hear more about, feel free to send an email to us, and we'll do our best to make it happen. So it gives me great pleasure to welcome Dr. Holly Kordasiewicz from ISIS Pharmaceuticals. Holly is currently an assistant director at ISIS, which you'll come to find out is if you're not already aware, is a biotechnology company focused on RNA targeting therapeutics. She obtained her doctorate degree at the University of Minnesota in neuroscience, where she was studying the molecular mechanisms of polyglutamine toxicity. She completed her postdoctoral training under Dr. Don Cleveland at UC San Diego, where she actually started working with ISIS developing this ASO therapy for HD. And since November of 2011, she's been working as a scientist at ISIS. Also joining us from ISIS is Christina Lemonitis. Christina's been at ISIS for over 20 years now, where she started as a scientist, where she was characterizing how these antisense oligonucleotides worked. At ISIS, she kind of she was given the opportunity to combine her role as a scientist and her nursing background to start up ISIS's first patient advocacy program to support the development pipeline. As we'll come to find out, ISIS has pioneered antisense technology and has a robust pipeline of nearly 30 different antisense drugs in development, and they plan to add 3 to 5 compounds per year. Hopefully, one of them will be targeted towards Huntington. So without further ado, I introduce you to, and we'll turn it over to Holly and Christina to present to us an update on their antisense oligonucleotides. Okay, so hopefully everyone can see this.
Hello, are we all set? Hello, everyone. Thank you, George, and thank you to everyone listening for giving us the opportunity to speak to you about our HD work. So I'm Holly, and I'm here with Christina. Hi, good morning, everybody. And we're going to talk to you today about the HD program at ISIS Pharmaceuticals. So first, just a little bit about ISIS. So ISIS specializes in antisense toxicology, or as we like to call our antisense oligonucleotides, ASOs, or antisense drugs. And ISIS has been working with ASO technology, developing drugs to help patients, particularly those with rare genetic diseases, since the 1980s. And as you can see, ISIS works in many different therapeutic areas, and this is a list of our programs here that are currently in clinical development. And one in particular that we're extremely excited about is in our cardiovascular program, and that is where we just received approval from the FDA for our ASO Kinamro. And this is an ASO to treat patients with homozygous FH, which is a fatal disease that causes astronomically high cholesterol levels that leads to an early cardiovascular death. But I'm sure everyone listening today is most interested in our neuroscience program. And for those, we have ASOs in 2 active clinical trials for 2 different diseases. One therapy is to lower TTR, which is a mutant protein in an inherited form of transthyretin amyloidosis, or familial amyloidosis disease. And the second is an ASO to treat children with SMA, or spinal muscular atrophy. And spinal muscular atrophy is an inherited neurodegenerative disease, and it's the leading genetic cause of infant mortality. And so we have a lot of things going on with ASOs, and we're hoping that, as George mentioned, HD will be on this list very soon. So now a little bit about ASOs. So ASOs are typically used to specifically prevent the production of an unwanted protein. In HD, that unwanted protein is mutant Huntingtin. So today I'll be going through the basic biology of how proteins are made, how the ASOs can be used to prevent the production of these unwanted proteins, and then finally the work that we've done to specifically lower the unwanted Huntingtin protein. So first off, our genetic material is encoded in DNA, and in cells the DNA holds the blueprint for the proteins that each cell will need to perform their different cellular functions. And the different proteins are made from different genes. So our DNA contains a number of different genes, and typically each one of these genes encodes for a protein. So one way to think about this is that if DNA is a book, then the genes are the words. And so in a little more detail of how DNA is made into proteins, and this is a mechanism that's really important to understand if you're going to be thinking about how antisense drugs work. And so here on this slide, we're going to start at the gene level. So as I just said, or as I just mentioned, the DNA contains genes, and so then each one of these genes is then transcribed into something we call an RNA, and this RNA is then translated into a protein. So now in HD gene positive patients, they have a mutant Huntingtin gene. This mutant Huntingtin gene is made into a mutant Huntingtin RNA, and that mutant Huntingtin RNA is then made into a disease-causing mutant Huntingtin protein. And it's largely believed that it's this mutant protein here that's what's leading to the neuronal death and disease. And now ASOs work by binding to the RNA, and this is the middle step in the process of how genes become proteins. And this is really where the specificity of our antisense drugs come in, because the ASOs will only bind to an RNA that has a matching sequence. So in our case, we've made ASOs that match the Huntingtin RNA sequence, and the binding of the ASO then leads to a degradation of the target RNA. So if we no longer have the RNA, then we prevent the translation event that happens, and then we decrease the levels of the protein. So for the Huntingtins, we have our ASO that binds to the Huntingtin RNA, so then that leads to a decrease in the Huntingtin RNA and a selective decrease in the Huntingtin protein. And then if there's less protein being made, then there's less of the bad protein accumulating, and then hopefully the idea is that there'll be less disease. Okay, so now to get into the data of exactly how these are working for our Huntingtin program. So to determine if we can really use these oligonucleotides to lower Huntingtin, we use mice that contain the full human Huntingtin gene, and therefore they make the Huntingtin RNA and the Huntingtin protein. And they have a disease that has some similarities to the human HD that we can measure in these mice and quantify the changes. So these mice were made by Dr. William Yang at UCLA, and he's let us use his animals for our studies. And in these mice, we introduced our ASOs that target the Huntingtin RNA, and we introduced the ASOs into the fluid that surrounds the brain. And then we measured the different levels of the Huntingtin RNA in the brains of these animals. So in this experiment, we treated the animals for 2 weeks with our oligonucleotide, and then we collected the brain tissue from the animals at different time points after our treatment, and we looked at the RNA levels. So in the blue line here, this is the RNA, the Huntingtin, the human Huntingtin RNA levels from the animals that received the ASO treatment. And these animals here in black, this black line is denoting the animals that they only received saline. So saline is what we call the vehicle solution. It's just the buffer that we put our ASOs in. And so hopefully what's clear to everyone is that when we end the treatment, the 2-week ASO treatment, the Huntingtin RNA levels are down. And the really exciting thing that came out of these studies is that this ASO treatment for only 2 weeks lasts for a couple of months. And then as expected, because the RNA is down, the protein is also down. So here you're looking at something we call a Western blot, and this is actually looking at the Huntingtin protein itself. This top band here, hopefully you can see there are 2 little bands here. This top band is the mutant Huntingtin, and this bottom band is the normal Huntingtin. And so this first lane is the animals that weren't treated with our oligonucleotide, and then here are all the animals at different time points that were treated with our oligonucleotide. You can see right at the end of treatment, the Huntingtin proteins are coming down, both the mutant and the normal. 2 weeks later, they're both clearly down, and then they stay down. And it's not here until the end that the levels come back up. And so what this is telling us is that after a 2-week treatment, we're having a really long effect of our oligonucleotides, and that we're lowering both the RNA and the protein, which is what we would have hoped to do. So now that leads to the big question. Does lowering Huntingtin with these anti-oligonucleotides have an effect on disease? And I'm sure, as most of you know, there's been a lot of other previous research that have shown that lowering lowering the mutant Huntingtin by different genetic methods has a beneficial effect on disease. But we wanted to see if we could have that beneficial effect with our oligonucleotides. And the answer is that in mouse models it does. So here is an example of motor coordination, and these are in, again, those mice that have the human Huntingtin gene, the mutant Huntingtin gene, and these mice are less coordinated than a mouse that doesn't carry the mutation. And if we treat them with an oligonucleotide for 2 weeks to lower the Huntingtin, their motor coordination improves. And let me walk you through the graph now. So here, this is a test where the mice are placed on a rotating bar, and the bar rotates faster and faster with time. And we measure the time it takes for the animals to fall off the bar. And you can kind of think about this as like a lumberjack running on a log. And so the longer they stay on the bar, the better they're doing, the more coordinated they are. So if you look here at this black line with the open squares, these are the animals that don't carry the Huntingtin mutation. So these animals, they learn the task over time. So this axis is telling us over time, and this is telling us how good they are at the task, so how long they can stay on that bar. So they get a little bit better as they age, but then they stay pretty flat for almost the year that we measured them. So they're pretty good at the task. And here, this solid black line at the bottom, this is the animals that contain the Huntingtin mutation. So these animals, as you can see, they're not very good at this task, even from the start of the study. So they're not very good. They do get a little better as they get older, but then they start getting worse. And they get a little bit worse as they age. And they're never as good as the animals that don't have the Huntingtin mutation. However, when we treat with our oligonucleotide, and we only treat with the oligonucleotide for 2 weeks, like I had showed you in the previous study, so right here in pink it's just showing you the window where we were treating them. And then if we follow the animals over time that receive the oligonucleotide, they start to get better. It takes them a while to get better, about 2 months, but once they're better, they stay better. And they stay better than the other Huntingtin mice that didn't receive any drug, and then eventually the drug begins to wear off and they start getting sick again. And so this is telling us that suppressing the mutant Huntingtin will improve the motor coordination in the HD mice. Right. So in similar studies, where we suppress the mutant Huntingtin with ASOs in either this mouse model or 2 other mouse models, we have seen in the ASO-treated animals a decrease in the loss of brain mass and improvement in motor coordination, as I just showed you in that graph. We've also seen an improvement in motor activity. So for motor activity, the HD mice we use are normally not very active, but after we treat them with our ASOs that lower the Huntingtin, their activity levels turn out to be very similar to the non-HD healthy mice. And the ASO mice were also less anxious than the Huntingtin disease mice that didn't receive the ASO treatment. And we believe that we're having such a broad effect on the disease, at least in these mouse models, because we have an on-mechanism therapy, meaning that we're targeting specifically the Huntingtin gene product. And by doing so, we're really getting rid of the source of the disease. And by getting rid of the Huntingtin RNA, we're getting rid of the Huntingtin protein, and then also many of the bad things it does in the brain. So most HD patients, they have 1 normal Huntingtin gene and 1 mutant Huntingtin gene. And you may have heard that there's a large debate in the HD research community whether it's necessary to selectively lower mutant Huntingtin without lowering the normal Huntingtin. And I think we agree that it would be best to selectively lower the mutant Huntingtin protein. However, is it really necessary to selectively lower the mutant Huntingtin protein? To selectively lower the mutant Huntingtin protein. So in the motor coordination study I just showed you, we were lowering both the mutant and the normal Huntingtin together, and we had a clear benefit in the mice. To determine if we would have had a better result if we would have just lowered the mutant Huntingtin without changing the normal Huntingtin, we designed an ASO that selectively lowered the mutant Huntingtin without altering normal Huntingtin in these mice. And here is the Western blot, just like the one I had previously showed you, confirming that we're just lowering the mutant Huntingtin alone. So these are the two bands. This top band is our mutant band. This lower band is our normal band. These are the animals that just received the saline vehicle. These are the animals that received our mutant Huntingtin ASO. And hopefully, as you can see, this top band is gone and the bottom band isn't. So we're, in this case, we're selectively lowering the mutant Huntingtin. And then, oops, sorry, the slides aren't advancing. There we go. And then if we do the same study that we had done previously, where we look at the motor coordination in these animals, and you see that here is, again, these are our back HD mice. So this, I've simplified the graph, so we're just looking at the animals that have the Huntingtin mutation. And these are the animals that were treated with our vehicle, and they get a little bit worse, as I showed you before. These are the animals in blue that I'd previously shown you that are treated with our total Huntingtin silencing oligo, and they get better. And then if we look at our oligo that just silences the mutant Huntingtin, they also get better. And the really remarkable thing is that they get better to a very similar effect, that we don't seem to be having a different effect if we lower just the mutant or if we lower the mutant and the normal together. And so because of this, we're working to take this total Huntingtin approach forward into patients. So although we haven't seen any detrimental effects from lowering the normal Huntingtin with the mutant Huntingtin, we are moving this total approach forward. We have alternate programs that are designed to explore our ability to selectively lower the mutant Huntingtin. And these are the SNP approaches that have been pioneered by the chemists here at ISIS and with Dr. Hayden's group at UBC, as well as the CAG targeting approaches that we are working on in collaboration with Dr. Corey at UT Southwestern. And so I've been talking a lot today about our first approach, which is the total Huntingtin approach. And the benefit of this is that with one ASO, we could treat all HD patients. And we have quite a bit of mouse data to suggest that this would be beneficial. And other scientists working with different gene therapies have been working on this too, and they've come to similar conclusions. However, for the mutant-specific silencing approaches, the benefit of these is that we would only be lowering the mutant Huntingtin and leaving the normal Huntingtin alone. And so I'll walk you briefly now through these other two alternate approaches. So here is a cartoon showing you what our non-allele selective ASO is doing. It's binding to both alleles. So just to remind you, patients typically have a normal Huntingtin allele that isn't expanded. It has a normal size CAG tract, and one with this expanded CAG tract. And this is the Huntingtin mutation. And so when we have our oligo that just binds Huntingtin, it'll bind both of these and lower both of these genes. Now, with our SNP targeting approach, our SNP targeting approach is targeting these little things that we call SNPs. They're single nucleotide polymorphisms. And geneticists have identified a number of these that are found in linked to the CAG expansion. So here you can see there's this little green dash, and this is denoting a single base change that just happens to be linked with the CAG expansion, and the most that they've been found is in about 50% of HD patients. So here on the normal allele in this particular patient example, they have a different base here. And so because this base is different on the expanded and the mutant, we can put our oligo right across from this base. And as I told you earlier, the oligonucleotides need to match the sequence, and if they don't match the sequence, they don't work. And so this is only because it only matches this sequence. It's only going to lead to the degradation and cleavage of the mutant, but it's not going to lead to the degradation of this one. Now, the benefit of this, as I said before, is we're only going to lower the expression of the mutant Huntingtin. The downfall is that to treat everybody selectively, we'd need multiple oligonucleotides because not everybody carries these SNPs linked to this CAG expansion. Now the final approach, and it's another allele selective approach, and this one would treat all of the patients, is one where we actually bind our oligonucleotides right to the CAG tract itself. And so because the CAG tract is expanded, we can get more of our drugs to bind to the expanded ones than they do to the normal ones. Now in this case, we don't want to use the RNA degradation mechanism. We can change our oligos so that rather than degrading the RNA, they'll just prevent the production of the protein. So the protein is made by machinery that runs across the RNA like this, and it'll get stuck and stop right here. And the more oligos that bind, the more stopping that's going to happen. So the less of the mutant Huntingtin protein that's going to be made compared to the wild type. And so this third approach is really exciting, and it's a really neat mechanism because it's kind of a different way to use oligos than what we traditionally do. But it's a really new mechanism, and so we're working now to optimize this type of delivery to get drugs into the brain. So it's further behind than our other approaches because it's not something we typically do. So there are benefits to each of these different approaches, and we are actively working on all of them. And today I have been focusing on our total Huntingtin approach since it is the most advanced of our programs. And on the previous slide, I've included references where you could read more about these different approaches. And I'd also like to refer you to that all three of these approaches have been reviewed very nicely by the HD Buzz guide. And you can find links to the HD Buzz website on the HDSA website. And they'll explain these in a lot more detail. So today I've been talking a lot about the preclinical work that we've been done in mice. And now that we're confident that we can clearly help the HD mice, the next step is to scale all this up for treatment in people. And one of the first questions that we need to answer when moving from mice to people is determining how to dose. And this is getting into one of the really exciting things about ASOs. And it's that they can suppress the gene production, the gene product production in a dose-dependent manner, meaning that you can give more or less drug if you want more or less Huntingtin reduction. And this is a really important point to make because I think there has been some confusion in the past, and that's the fault of us scientists who may have heard us use terms like Huntingtin knockout or gene silencing, but in reality it's not knocking out or silencing of a gene. It's a Huntingtin lowering or a Huntingtin suppression approach because we won't be completely abolishing Huntingtin. Rather, we'll just be lowering it. And the ASOs can lower it to different degrees depending on how much of the ASO you give. And here's an example of that. Here we've taken our ASOs again into mice, and these are the mice that have that human Huntingtin. And we give the ASO at different doses. And in this study, we weren't using any infusions or anything fancy. We just gave a single injection right into the fluid that surrounds the brains of these animals of our ASOs at different doses. So these are the different doses we used. And then we collected all the tissue 2 weeks later. Then we determined the levels of the Huntingtin RNA. So this is how much of the Huntingtin RNA is left in these brains. And we looked in 2 different brain regions. This is the data from the cortex, and this is the data from the striatum. And what you can see, hopefully, is a very clear dose response, meaning that if we want a 50% reduction in the Huntingtin RNA, we give 100 micrograms a dose of drug. If we want a 75% reduction, we give 300 micrograms. And this property of ASOs is really nice because we can give different amounts of drug to get really different effects. And this is different from some of the other gene therapies out there, which are really all or none. So it gives us the opportunity to titrate the dose, and this way we can make sure we lower Huntingtin enough to have an effect on the disease, but to still be safe. So in patients, we would like to decrease mutant Huntingtin throughout the brain. And as you know, HD is a very complex disease. There's many different brain regions involved, and to affect as many of the different aspects of the disease as possible, we want to get the ASOs to as many different brain regions as possible. However, the brain has this natural barrier to protect itself, and many things can't cross this natural barrier. It's called the blood-brain barrier, and at this time, our ASOs don't cross this barrier. So injecting them intravenously or taking them as a pill isn't going to get them into the brain. So to get them into the brain, we inject the ASOs into the fluid that surrounds the brain and the spinal cord. And this fluid is called the cerebrospinal fluid, or CSF. And the route of administration, this type of delivery of injecting it right into this region, is called intrathecal, or an IT injection. And so for this type of injection, a needle is placed right into the intrathecal space, and then the drug is injected directly into the CSF, the cerebrospinal fluid, which you can see here in blue, bathes the whole brain. And then we do this rather than directing it directly into the brain tissue itself, because the CSF bathes the brain, then because we put our ASO into that CSF, it'll be bathed the brain, and we get a better distribution to broader distribution to more areas and get more Huntingtin reduction in more different areas of the brain. And another reason for going IT other than other types of delivery to the brain is because this is a somewhat routine type of injection. It's done for chemotherapeutic agents and things like painkillers. And it's something you may have heard of. It's a little bit like a spinal tap, if you've heard of that. And it's also a route that we have experience with because our other CNS programs that are now in clinical trials in patients are using this intrathecal type of delivery. So in mice, I've shown you that when we introduce the drug into this fluid that surrounds the brain, we can get good reductions in Huntingtin mRNA and protein. But mice are really small, and so we wanted to see if we could get good reductions in the Huntingtin in a larger brain. So we've also done studies in monkeys. So in monkeys, we injected ASOs into that intrathecal space, just like we do in humans, into the fluid that surrounds the brain, and then we determined where the ASOs went in the brain. So in these pictures, you're looking at pictures of a monkey brain, where the brown staining is our oligonucleotide. And here, if you look at the close-up, zoom up to some of these different regions, this blue staining are the different cells within the brain. So you can see that lots of the different cells and areas have our oligonucleotide. And just to orient you, these are something called coronal sections. So where the top of the section, this would be the top of the head, the bottom of the section, this would be the bottom of the head. And this is near the front of the brain, so near the forehead, and this is near the back of the head. And so as you can see, even after introducing the drug into the intrathecal space, way down at the spinal cord, we're getting drug into regions all the way into the front of the brain. And so we are getting good distribution in these monkeys. But we also wanted to look at the Huntingtin knockdown, and since this oligo we put into monkeys matches the sequence of the monkey Huntingtin RNA, we can look at the monkey Huntingtin RNA levels. So here on this axis, you're seeing the monkey Huntingtin RNA levels. And so in this case, the vehicle is in blue, so this is the 100% level, and then how much it's knocking it down. And hopefully, what you can see is that in the spinal cord and cortex, we get really good knockdown, and then with a little bit less knockdown in the more deeper brain structures. And another thing that we've done is one thing that seems to be really consistent between all the different species that we've looked at, so both in mice and rats and in monkeys, is that our ASOs last for a long time in the CNS. Meaning that weeks after treatment has ended, we can still see a suppression in the Huntingtin RNA and in the protein. And so I've shown you this in mice, but here is a similar study in monkeys. We treat the monkeys with the ASOs, and then we collect the tissue at different time points after injection and look at the Huntingtin RNA levels. So these are the different time points in 3 different brain regions. You can see that the Huntingtin levels stay down, and then they start to come back to normal about 2 months after treatment. So this is good news for the patients because it means that we have a really long half-life of the drug and that the injections will need to be less frequent. And that's important for a disease that goes on for a really long time like Huntingtin. So to summarize everything I've spoken about today, is we hope to use ASOs to decrease the Huntingtin RNA, which in turn will lead to a decrease in the Huntingtin protein, and that we think that if we can do this in patients, that we have a real shot at ultimately decreasing the disease. And the big question, I'm sure, on everyone's mind is when. And we plan to identify a development compound this year. And once we have the development compound, it takes a little less than a year to complete the safety studies in animals required by the FDA before we advance the drug into patients. And these studies are called Investigational New Compound Enabling Studies, or IND Enabling Studies. And if everything goes well in the IND Enabling Studies, then we're allowed to proceed into the safety studies in patients. And this first stage of clinical trials are the safety studies in patients, or the phase 1 trials, and we plan on being at this stage by the end of the year in 2014. So I'd like to thank you all for your time, and a thank you to all of our colleagues, both in academics, in industry, and at CHDI, all of whom continue to work very hard on this project. And a special thank you to all the families and patients out there who continue to support our work and participate in natural history studies and clinical trials. And so we're happy to take any questions now.
Great. Thank you very much, Holly. We will just go to see if anyone has any questions. Just give us 1 second here. I'll start while we pull these up. Holly, I'm really intrigued by the looks like it's really robust spread in the cerebellum, yet it appears to be resistant to Huntingtin knockdown. Is that something you could comment on?
Yep. Yeah, so that's a really great observation. So that's something we see consistently, both with Huntingtin and a lot of our other targets. It seems that the granule cells, which make up or the most abundant cell population in the cerebellum, are resistant to our antisense oligonucleotides. We don't know why. They seem to be the one clear cell population, the CNS, that doesn't use the RNase H active mechanism that our oligonucleotides use. However, in the cerebellum, we do see very clear reductions in Purkinje cells if you do things like a C2 hybridization. But when you take that big piece of tissue, the Purkinje cells make up a small population of that percentage of that piece of tissue.
Thanks. Okay, so we've got quite a few questions here. Let's see. We have a question here. When you start, if early enough, would it be possible to maintain performance to the sample population? Not exactly sure. Let's move on to this question. What stage of HD were the mice and monkeys in at the time of injection? And do you think you'll have the same outcome in advanced stages and beginning stages of humans?
So that's an excellent question. So I should clarify. So the monkeys weren't Huntington's disease monkeys. Those were just regular monkeys. They didn't have any disease. We just targeted their normal monkey Huntingtin. So we didn't do any proof of concept behavior studies in those monkeys because they were just normal healthy animals. Now for the mice, we have done those exact studies that you're asking about. So we treated animals when they were 2 months of age, which is they don't have all the aspects of Huntington's disease. They're just starting. They're right around what you might call phenoconversion. So they're just starting to show the symptoms. And then, of course, they're going to get worse as they age. When we do them there, we have a really robust reversal in disease, and that's where we get our strongest effect. When we treat animals later, so we've treated animals when they're 6 months of age, when they have a very clear phenotype, and those animals, we also see an improvement, but it's not as robust. So it seems the later you treat in the disease, the less of an improvement you have. And that kind of makes sense. So if you think that we can have an effect on the cells that are still there, they're just not performing properly, but once the cells are gone, they're gone, and we can't help them.
Holly, you have a really robust effect around 4 to 6 weeks post infusion of your ASOs. Have you ever tested earlier, like in neonate animals, and seen if there's any kind of detrimental effect? Because there's, it's postulated that Huntingtin may play a developmental role, an important developmental role early.
No, so we haven't done anything in really neonatal animals. The earliest we've ever gone is 8 weeks.
8 weeks.
Or I'm sorry, 4 weeks is the earliest we've gone. So it's after the animals are already weaned from their mothers, and so they're considered adult mice.
Okay. Another question here, and it is, what are the size of the oligonucleotides?
So the oligonucleotides, the ones that we're typically using for this are 18 to 20 bases in length.
Great. There's a question, could you clarify what the wild-type Huntingtin gene means?
Oh, yep, so wild-type is just a normal Huntingtin gene. That's something us mouse geneticists like to use that phrase a lot. Wild-type just means the normal Huntingtin that would be found in the wild. So in patients, you can think of just an unexpanded Huntingtin. Yeah, that's a mouse term that sometimes we carry over. Sorry about that.
It's okay. Sorry, let's see. Where are we? What do you anticipate would be side effects of using the treatment to reduce the total Huntingtin gene and not just the mutant?
So far we haven't seen any detrimental effects. So there have been reports as you mentioned, in development, but we do not plan to go developmentally. We plan to treat adult patients, and so far in the adult animals we haven't seen any side effects from lowering the normal Huntingtin. So far we hope that's going to stay the case. Of course, we'll have our safety studies for our IND-enabling tox studies, and that'll really determine how safe everything is, and if we can take it into patients.
Great. There's a technical question for you, Holly, for the scientists out there. Was the Alzit type pump used to infuse the ASOs for the 2-week time point, or did you do direct ICV injections?
No, they were all the osmotic pumps. So at the time, we didn't realize, the Huntington program has actually taught us a lot about our oligonucleotides. At the time, we didn't realize that the half-life of the drug was so long. We based those studies on what we knew when we deliver the drug systemically. And so we were using the pumps to try to get a lot of the drug in there. And so those were all the Alzit pumps. But since then, we found that the ICV bolus works just as well. We can get just as much drug in, and because it lasts so long, it's an easy way to deliver it to mice. So now we're doing everything with those ICV bolus studies without using the pump.
Great. This is a more of a broad question. Is there anything that the families can do to help ISIS with their efforts?
Not ISIS particularly, but I'd say in general, the biggest thing for us in our next hurdle going forward is what markers and what things to look at in the clinic. And so anybody that can participate in these natural history studies so that we can understand really how the disease course is progressing and how it's following is going to be a huge help to us. Because right now, we can measure, we have all these great endpoints that we can look at in mice, so like I showed you that little rotarod test, but we can't ask patients to run on a log. So they have different things that are going on now. CHDI is doing a lot of that work with things like Track HD and Predict HD, and studies that are really following how the disease changes over time and how we can quantify that, so that when we do begin our clinical trials, when we want to see if we're having a benefit, we know that we're looking at the right endpoints and the right markers, and that we know what the natural progression is so that we can see if we're stopping that natural progression. Because HD, it might not seem like it to the patients, but it really is a slowly progressing disease, and so that you sometimes have to look over a couple years to see changes. And so that's why we need to know exactly what are the best things to look at, what changes the fastest. So participating in any of those kind of studies is the best thing that I think you can do for us right now.
Great. Thank you, Holly.
It's really important going forward.
Totally agree. There's a question, if you could, based on what you've seen so far in the mice, could you speculate on, or is it too early to tell on how often your ASOs would have to be administered to humans to maintain the same Huntingtin lowering effect?
Yeah, so that's a great question. So actually that's what we're working on modeling now. And so we're thinking it's possible that we maybe even go as low as quarterly, so 4 times a year. But of course, none of that's set in stone. We're doing another monkey study where we're going to be looking at exactly what our lead compounds are doing and how our lead compounds, which are one of them is going to become our development candidate, how those are lowering Huntingtin and in the larger brains, and then using that to model how often we would need to dose in patients.
And in a clinical trial aspect, if you look at the SMA study that we're doing, that's another predictor, because in those patients, that's also an IT delivery. And that we're finding out that we'll probably only need to dose between every 6 to 9 months, or maybe 6 to 12 months. Mm-hmm. So we are learning about the delivery and how often we'll need to do drug delivery in some of the other studies that are ongoing.
Yep.
It's actually a kind of like a drug delivery question, technology question here for you. Where did it go? Hold on a second. Sorry. Yeah. How far along are you in developing pre-ASOs or carriers of the ASOs that can be delivered systemically and can actually cross the blood-brain barrier and access the central nervous system? Is that type of work that you are engaging in? This, looking for alternatives besides the intrathecal administration.
Yeah, so that's great. So we don't have any active programs currently ongoing right now, but that's something we have explored quite a bit in the past, and are talking about working with other collaborators to do that type of thing. But it's not something we have an active program going on right now, but it's definitely something we're interested in.
Great. Let's see here.
But just to kind of delivering systemically would be nice, but because of our long half-life and because we do think we're getting a good distribution IT, we think we don't need to wait for anything like that to start our clinical trials, that we can take these forward IT.
Great. So could you describe the stage of progression that the first safety tests will be conducted on for a person? Is it early, middle, late, or something else?
So we don't have anything written in stone, but I can just give you kind of the general thought processes that are going on. And we're still talking to a lot of the key opinion leaders in the Huntington's field, and so things can definitely change. But we think it needs to be symptomatic patients so that we can really see the progression of the disease and the changes that are going to happen. And so we think that we'll do our safety studies in the same patient population that we'll be doing our phase 3 proof of concept studies in, which would need to be patients that have some type of phenotype that we can follow and see if it changes.
Mm-hmm.
So that's the latest that I've heard.
So question here about off-target effects. And are you worried about off-target effects, or is this not so important because of the transient nature of the ASO therapy?
No, so we do think about off-target effects. So all of our lead compounds are selected because they don't match anything else perfectly in the human genome. And anything that they match even remotely closely, we check. And we test to make sure that they aren't down. And if a compound is having an off-target effect on another gene that would be found in the human brain, then it would be canceled out of the running. So that's one of the good things about oligonucleotides, and that's one of the things that takes a long time in our screening process, is we can make oligos against the entire Huntingtin pre-mRNA. So that's a ton of real estate. So we can really select and throw out compounds that are having any of those off-target effects.
Great. Another question about blood biomarkers, or peripheral biomarkers. Are there any biomarkers that you, at ISIS or CHDI, are going to be planning on using to assess how well your therapy is working in humans, or are you still in the process of developing those biomarkers?
So we're still definitely developing them, and that's where our big force and our big push is right now. One thing, blood biomarkers isn't the best for us because it would have to be something that is secreted from the CNS into the blood. Because our oligonucleotides are delivered at such a low dose, because we're putting them directly into the cerebrospinal fluid, the amount that leaks out of the actual active oligo that gets into the blood is so low that it's not going to lower anything systemically. So it's not going to lower things in your blood cells or in your liver or in any other systemic targets. And so because of that, it has to be something that came from the CNS, because that's where we're having our target knockdown. And so because of that, most of our markers that we're looking at are CSF biomarkers. And so we do have a really exciting study that we're doing with CHDI and Kitamed, where we're looking at things that are secreted from neurons into the CSF and seeing if those are changing. And we're also looking at the actual Huntingtin protein itself in the CSF to see if we can detect that and see if the Huntingtin protein itself is changing. And then there's also a lot of work that's been going on in Don Cleveland's lab at UCSD, where they're doing big gene screening array studies and RNA-seq studies looking at all the different genes that are changed after we lower the mutant Huntingtin in diseased animals, and seeing if any of those things that are changed, if those are things that are secreted or can be found in the CSF, like, for example, BDNF.
Great. Last I heard that detection of Huntingtin in the CSF, the cerebral spinal fluid, is very, very difficult. Are you seeing progress in that area, that you're able to detect it with current technologies?
It is. So not for Huntingtin, but we're working on with a number of different collaborators and companies who specialize in optimizing those assays for our SMA program. And so the more that we learn about that and how they can do this very, very low level detection, that we're hoping that we can use what we've learned from our SMA work and apply that to our Huntingtin program.
That's great. I think you talked a little bit about it. You showed it in the chart, but are there ASO clinical trials that are farther along for other diseases? Maybe more particular, we saw the recent approval of the cardiovascular ASO, but are there ASOs in the neurodegenerative field that are further along than the ASO program for Huntingtin?
Yep, so the SMA drug, so the spinal muscular atrophy, this is an intrathecal delivery of a drug into children. So these are children who have a neurodegenerative disease, and it's a fatal neurodegenerative disease. And this one, we completed our phase 1, and it's actually going into phase 2. So I think we started the phase 2.
It's in phase 2, and it'll actually go into phase 3 next year. And then the other program that's actually advanced is TTR. Our TTR program initiated phase 3 studies just about a month ago, a few weeks ago actually. So that is not an IT injection, that's just a sub-Q injection, but that is one of our more advanced neurodegenerative programs.
I have one more question. I know you showed really nice data for the wild type or the non-Huntington's monkey in showing the spread. Are there plans at ISIS and CHDI to test your oligonucleotides in a large animal such as a sheep or an HD monkey to test for effectiveness?
Yeah, those animal models are so cool, but no, right now we haven't been planning on doing that because our oligos. A lot of our human lead candidates cross-react. They're identical to the monkey sequence. Using a wild-type monkey, we think will give us the readout without having to go into specifically the diseased animals.
Great.
But it is a really cool idea.
I think are we at the end? I think that's
nothing after that.
Okay. Great. I mean, I think that's most of the questions. A lot of the questions are I've seen there are, how can we get involved and how can we get, everyone is very excited about participating in these potential clinical trials, and I would say just stay tuned, and as these things mature, you'll hear about them at HDSA. We'll make sure the families and caregivers are all well aware, and just participate in, make sure you participate in the observational trials such as Enroll HD. Also keep looking up for sign up for hdtrials.org, which will notify you of new clinical trials in your area as they become available, as well as, I think Holly alluded to, clinicaltrials.gov, where you can research any and all of the different clinical trials that are ongoing across the country in Huntington's disease. So if there are no other questions, I just want to thank Christina and Holly for an outstanding presentation. I really appreciate you guys coming in early and on the West Coast to present to all of us. And as I said, we will have recorded this, and for those interested, can tune in on our website probably within a week to see an archived version of this presentation. So thank you very much, everyone.
Thanks
Thank
for
you for
having us.
having us.
Thank you.